There is a swarm in the Southern Ocean so dense that a ship's instruments read it as solid ground. It is made of animals you could hold in the palm of your hand. Most people file them under plankton, drifting, passive, replaceable, food for whales. That answer is true and almost useless because it explains nothing about what these animals move. What leaves their bodies matters more than what eats them. Remove them, and the failure does not start with whales. It starts somewhere you would never look, and we still cannot say how many of them are down there. The screen shows a seafloor that should not be there. A research vessel is working south of the ice edge, echo sounder pinging downward into water more than a kilometer deep, and the return comes back wrong. A hard, dense band rising off the bottom trace. On some displays, it looks like a submerged mountain. On others, it looks like the ocean floor has lifted several hundred meters in the space of a few minutes and is now moving under the hull at walking pace. It is not rock. Nothing down there is solid. What the ship is reading is a mass of animals packed so tightly, in such numbers, that sound bounces off them, the way it bounces off stone. Now go down and look at one. Under a macro lens, on a fingertip, a single Antarctic krill is a small, translucent thing about the length of your little finger. You can see straight through parts of it. The shell is glassy, faintly speckled with red-orange pigment. Black compound eyes sit on short stalks, disproportionately large, the way eyes get in animals that have to see in bad light. Along the underside, a row of thin paddle-like limbs beats in a steady, rippling wave, front to back, and does not stop. That is the first thing worth noticing. It does not stop. We call these animals plankton, and the word does real damage to how people imagine them. Plankton is a category about movement relative to currents, organisms that cannot outswim the water they live in. It says nothing about whether an animal is passive. Somewhere along the way, the word collapsed into an image. Dust, drifting, at the mercy of the sea, closer to a floating particle than an animal. Watch one for 30 seconds and that image falls apart. Krill are dense, slightly heavier than the water around them. With no gas bladder, no oil float, nothing to hold them up. Stop swimming and you sink. That is the baseline condition of this animal's entire life. Those beating paddles underneath are not a nervous twitch. They are the thing keeping it in the water column at all, running constantly through the polar night, through winter, through storms, for years. Think about what that costs. Every second of existence has a running fee attached to it. Most animals get to rest by standing still. This one does not have that option. And the consequence shows up later in ways that reach much further than a single body. Because an animal that must burn energy simply to stay at a given depth is an animal under permanent pressure to find more. Hold that thought. It becomes the engine for almost everything else. Now pull back out to the swarm, because a single krill is not the interesting scale here. From the air, a large swarm shows up as a reddish-brown stain on the sea surface, spreading out under the light like spilled rust. It can run for kilometers. It moves. And when a ship crosses one and the sonar paints it in cross-section, you see something that a cloud of drifting particles has no business having. Edges. Not a fuzzy gradient thinning out politely at the margins. A boundary. Dense inside, near empty water immediately outside. The transition happening across a distance you could swim in a few seconds. Inside that boundary, the animals are packed at densities that in places run to thousands, sometimes far more, in a single cubic meter. A volume roughly the size of a household washing machine. Reach into it, and you would close your hand on living tissue rather than water. And they are lined up, inside a swarm, krill orient. They face broadly the same direction, hold rough spacing from their neighbors, adjust when a neighbor adjusts. It is a school, in the sense a herring school is a school. A coordinated structure held together by animals reacting to each other continuously. Currents cannot do that. Currents smear things out. Currents produce blurred patches, not defined bodies with membranes of empty water around them. So the swarm is not something that happens to krill. It is something krill do. And it is not one shape. Swarms have been recorded as compact balls, as long ribbons drawn out along a current, as flat sheets spread across tens of meters of depth, and as columns standing upright in the water. Some are small enough that a single whale could take a serious bite out of one. Others run so large that a ship needs hours to get across. The animals inside are not idle either. They keep feeding while the structure holds, which makes a swarm three things at once: a defense, a feeding aggregation, and a moving front that strips the water it passes through. Which raises the obvious question: why bother? The standard answer is safety in numbers, and the standard answer is right. But it is worth being precise about the mechanism, because the mechanism is what breaks later. When you are one animal among millions, the arithmetic of being eaten changes. A predator that arrives and takes 100 individuals has taken a rounding error. Your personal odds improve enormously simply by being surrounded. Add to that the confusion effect. A predator trying to lock onto one target in a moving wall of identical bodies has a genuinely hard visual problem. And the calculation looks excellent. There is also the escape itself, and it is worth seeing in slow motion. A krill under threat does not flee forward. It flexes its abdomen and snaps its tail underneath in one violent contraction, firing itself backwards through the water at a speed that, scaled to its body length, would humiliate most fish. Then it does it again. In a swarm, one animal triggering that response once triggers its neighbors, and the reaction spreads outward as a visible pulse. A hole opening in the mass. The whole structure flinching away from the point of attack and closing behind it. Watch that on film and it looks like a defense that works. Here is where it stops working. Every part of that system, the dilution, the confusion, the backward flick into the gap, assumes a predator that hunts one animal at a time, something with jaws, or a beak, or a grabbing limb, that has to select a target and commit to it. Against a fish, against a squid, against a penguin. That assumption holds, and the swarm earns its cost. Against a mouth the size of a room, the assumption is worthless. A lunge-feeding whale does not select. It accelerates into the swarm, opens its jaw to an angle that looks structurally impossible, and takes in a volume of water that can exceed the volume of its own body. The throat pleats ballooning outward into a shape that has no business belonging to a vertebrate. Then it closes, drives the water back out through the baleen, and swallows whatever the plates held. Dilution does not help you when the predator takes the entire neighborhood. Confusion does not help a hunter that never aimed at anything. The tail flip escape fires you backwards into water that is already inside the mouth. The defense did not fail. It was solved. And then, in the 20th century, something else found the same loophole. Once you know that these animals gather themselves into dense, locatable, sonar-visible concentrations, the hard part of catching them disappears. You do not have to search the ocean. You have to find the patch, and the patch announces itself. Modern krill vessels can work a swarm with gear that pumps the catch aboard continuously without hauling the net, turning what would be a series of discrete hunts into something closer to a tap being opened. The thing that keeps an individual krill alive is precisely the thing that makes the population easy to remove. That is not irony. That is the shape of the problem, and it comes back with force much later in this story. So here is where the first piece lands. The swarm is not weather. It is not an accident of eddies and wind. It is an active structure maintained second by millions of animals that have to swim anyway, each of them making the same bet, that being surrounded is worth what it costs, which leaves the question that should be simple and is not. If these swarms are large enough to register as false ground on a ship's sonar, visible from an aircraft as a stain across the surface, dense enough to grab by the handful, then we ought to be able to say how much krill exists in the Southern Ocean. A rough number, an order of magnitude everyone agrees on. We cannot. The estimates span a range so wide that the low end and the high end describe two different oceans. And the reason for that gap is not laziness, and it is not lack of ships. It is that this animal has a set of habits that sabotage nearly every method we have of counting it. Start with the part that should be easy. We can find these swarms. We can photograph them from aircraft, map them with sound, scoop them out by the ton, freeze them, weigh them, count them under a lamp. A commercial fleet works them every season and knows exactly where to go. So the total ought to be a settled number by now, argued over the third decimal place, estaran decimal tsao printed in every textbook. Instead, the published figures for how much Antarctic krill exists have ranged over the decades from a few 100 million tons to several times that. Not a disagreement about precision, a disagreement about which ocean we are living in. That gap is the story. Because the reasons behind it are not administrative. They are biological, and each one tells you something about the animal that you would never learn from a photograph. Begin with the oldest method. Drop a net. Tow it through the water for a fixed distance. Count what comes up. Multiply out across the area. Simple, physical, hard to argue with, except a krill can see the net coming. Those oversized black compound eyes are not decoration, and the tail flip escape does not care whether the thing approaching is a fish or a metal frame trailing a cone of mesh. Tow slowly, and the faster individuals get out of the way. Tow at night, and you catch more, because visibility drops, and the animals near the surface are easier to surprise. Tow in daylight, and the same water can come back nearly empty. Not because the krill have gone, but because they have gone down, or sideways, or simply outmaneuvered you. So your sample depends on the hour, the light, the speed of the ship, the size of the mesh, and the swimming ability of the specific animals you happen to meet. Larvae slip through mesh built for adults. Adults dodge mesh built to hold them. Every one of those effects pushes in a different direction, and none of them are constant. Then there is the deeper problem, and it has nothing to do with gear. You are sampling a patchy animal with a method that assumes an even one. Think about what the swarm actually is. Enormous concentrations, then long stretches of water containing almost nothing. If your toe happens to cross a swarm, your number is astronomical. If it passes a kilometer to the side, your number is close to zero. Multiply either result across a survey area the size of a continent, and you get two completely different answers from the same afternoon of work, which is why sound took over. An echo sounder does not need to catch anything. It fires acoustic energy downward and listens for what comes back, and it does this continuously. Along every kilometer the ship travels. You are no longer sampling points. You are cutting a vertical slice through the ocean and watching it draw itself on a screen. This is the technique that produces those images of false ground rising off the bottom trace. It is a genuine advance. It also comes with a hidden step that most people never hear about. The echo sounder does not measure krill. It measures returned energy. To convert that energy into a mass of animals, you need to know how strongly one krill reflects sound. And a krill is one of the worst acoustic targets in the sea. There is no bone in it, no gas bladder, no air, no hard reflective surface. A krill is essentially a bag of watery tissue suspended in water, and the density difference between the animal and the medium around it is small. Most of the sound passes straight through. What comes back is a faint echo from a body that barely disagrees with the ocean. Faint echoes are workable. Inconsistent ones are not. Tilt the animal, and the echo changes dramatically. A krill lying broadside to the beam reflects differently from one angled nose down. And the difference is not a rounding error. It can be enormous. Now remember what a swarm does. It orients. Millions of animals aligned. And that alignment shifts when they feed, when they migrate, when something frightens them. Which means the same swarm, unchanged in every biological sense, can return a stronger or weaker signal depending on which way its animals happen to be facing when the ship goes over. Body length matters too, and so does shape, and so does the size mix of the population that year. All of it has to be estimated and fed into a model before the screen image becomes a tonnage. Change the assumptions in that model, and the total for the entire Southern Ocean moves. This has happened. Part of the historical spread between high and low estimates is not the ocean changing. It is our understanding of how a small, transparent body bounces sound changing. Then there is the question of what else is down there. Salps reflect sound. Fish reflect sound, some of them far more strongly. Other krill species live in the same waters. A dense layer on the screen tells you something biological is present, and it takes multiple frequencies, net verification, and experience to say what. Get that wrong, and you have counted the wrong animal. Now widen out, because the technical problems are only half of it. The Southern Ocean is not a study site. It is a band of water wrapping the entire planet, and a research vessel moving through it is a needle drawing a single thread across a bedsheet. Surveys run along transect lines, and between those lines is water nobody looked at. The largest coordinated efforts have covered a fraction of the total in a fraction of the year, in the months when a ship can safely be there. Which brings up the gap that matters most: winter. Ice closes over the areas where krill matter most at exactly the season that determines whether a generation survives. Ships cannot follow. Daylight collapses. Conditions become dangerous. Some work happens with moored instruments and under ice platforms, but nothing approaching the coverage the summer gets. And the summer is not when the hard part happens. We are best at observing this animal during the easiest months of its life. Depth adds another blind spot. For a long time, the working picture had krill living in the upper few hundred meters, and surveys were designed around that assumption. Observations since then have found them far deeper, down near the seafloor, in canyons and along the slope, well outside the layer being routinely counted. Nobody yet knows what fraction of the population uses that deep habitat, or when, or why. If a meaningful share of them are down there while the ship is measuring the top, the survey is not wrong so much as incomplete in a way it cannot detect. So the number is soft. Accept that. And something more important comes into focus. The absolute total was never the useful figure anyway. What actually matters is the direction. Whether there is more or less of this animal than there used to be, and where. That should be an easier question. It is a harder one. Krill abundance swings violently between years for reasons that have nothing to do with long-term health. Reproduction succeeds spectacularly in some seasons and fails almost entirely in others, driven by ice, food, and water conditions. A strong year class can dominate the population for years afterward, which means two surveys 5 years apart can differ by a factor that looks catastrophic or triumphant and mean nothing at all. To separate a real trend from that noise, you need decades of consistent measurement, taken the same way, across the same waters. And that is the uncomfortable part. Systematic large-scale work on this animal began long after industrial whaling had already stripped out most of its major predators, and around the time a commercial krill fishery was starting up. Whatever the Southern Ocean looked like before human hands were in it, no survey ever recorded it. The baseline is not a measurement. It is a reconstruction. None of this means the science is guesswork. It means the science is honest about the size of its own error bars, which is not the same thing, and the estimates that regulators use to set catch limits are built specifically to stay on the cautious side of that uncertainty. but it does leave us in a strange position. An animal, we agree, is holding up an entire ecosystem, and we cannot say to within a factor of several how much of it there is. So the counting can wait. There is a better way in, and it starts much smaller, with the question of how this animal makes a living at all. Because before krill can be food for anything, they have to solve a problem that sounds close to absurd. They have to eat an ocean that is almost entirely water and extract from it something dense enough to build a body out of. Take a bucket of Antarctic seawater in summer, the kind that looks faintly green when you hold it up, and let it settle. What you are holding is mostly water. The living material in it, the drifting single-celled algae that everything down here ultimately runs on, is present in quantities you would struggle to see if you spread it on a plate. That is the resource. Diffuse, microscopic, scattered through a volume so large, the numbers stop meaning anything. And out of that, an animal has to build a body dense enough that a whale can make a living eating it. This is the step everyone skips. We talk about the Southern Ocean food chain as though the hard part is at the top. The hunting, the diving, the enormous mouths. The hard part is at the bottom, somewhere between sunlight hitting a cell too small to see and a 100-ton animal filling its throat. Something has to do the concentrating. Something has to take energy that is spread across cubic kilometers and pack it into a form that can be swallowed. That job belongs almost entirely to one animal, and it does it with a piece of equipment that most people have never looked at closely. Turn a krill over. The first thing you notice is that it does not look like a shrimp underneath. The word shrimp gets attached to these animals constantly, and it sets up the wrong picture. Something scuttling along a bottom, picking at debris with small claws. Krill do not pick. Krill do not scuttle. What runs along the underside of the thorax is a set of 6 pairs of jointed limbs, held together, curved inward, forming a hollow cage that opens forward like a scoop. That structure has a name in the literature, and a much better description in plain language. It is a basket. And it is not solid. Look closer at the limbs, and each one carries a row of fine bristles. Look closer at the bristles, and each of those carries an even finer row of side branches. Then, on some of them, finer branches again. What starts as a set of legs resolves under magnification into a hierarchy of hairs on hairs, arranged so that the gaps between them close down to a scale measured in the small end of micrometers. Legs became a filter, not metaphorically, structurally. Now watch it work, and the elegance of it lands. The basket opens as the animal moves forward, drawing water in. Then the limbs close inward, and the water is squeezed out through the mesh, sideways and backward, while anything larger than the gaps stays behind. Trapped material gets swept forward toward the mouth parts. Open, close, push, swallow. Repeated continuously, thousands of times. The animal swimming and eating in the same motion because it cannot afford to stop doing either. Remember from earlier that a krill sinks if it stops swimming. Here is why that constraint turns out to be almost a gift. The propulsion it must pay for anyway is the same current that drives water through the feeding basket. The animal is not swimming and then feeding. It is swimming through its own food supply, straining it as it goes. You can see the result without any equipment at all. A krill that has been feeding well has a bright green gut, clearly visible through the transparent shell. A stripe of concentrated algae running the length of its body. Look at one and you are looking directly at compression in progress. Water in, cells out, packed into a line of pigment you could see across a room. Multiply that by the swarm. Millions of animals, in a defined volume, every one of them pumping water through a mesh filter all day, all night, through the summer bloom. A large swarm passing through a patch of productive water strips it. What goes in as invisible haze comes out as bodies. Dense, protein-rich, oil-rich, and crucially, big enough to be worth chasing. That is the function. That is what makes this animal irreplaceable. And it has nothing to do with abundance. Krill are the step where diffuse becomes concentrated. They are the ocean turning something unusable into something a predator can build a body around. Take that step out, and the energy does not disappear. It stays where it is, spread thin, in cells too small for anything large to gather efficiently. The sunlight still lands. The algae still grow. The energy simply stops being reachable. But no piece of equipment is universal. And this is where the elegance starts to show its edges. A filter has a mesh size, and a mesh size is a decision. Set the gaps too wide and small cells stream straight through, uncaptured. Set them too narrow, and the resistance to pushing water through goes up, which costs energy, which the animal pays out of the same budget it is trying to fill. Antarctic krill are tuned to a particular band. They are extremely good at capturing the larger phytoplankton, the diatoms, the armored cells that dominate the polar bloom when conditions are right. Those are the target. That is the shape of the tool, which means the tool has a failure mode, and it is not obvious until you ask what happens when the food changes. Warmer, more stratified surface water tends to favor smaller phytoplankton over the large diatoms. This is one of the broad expectations in ocean science, seen in warmer regions and increasingly a concern for polar waters, though how far and how fast it plays out in the Southern Ocean is still being worked out. Follow the logic anyway, because the logic is what matters. Shift the plankton community towards smaller cells, and the ocean does not become less productive. It can stay just as green. What changes is the particle size. And a filter built for one size class does not care how much food is present if the food no longer fits. An animal could be swimming through abundance and still be losing. That is a far more unsettling failure than starvation. Nothing looks empty. The satellite images still show a bloom. The system underneath simply stops transferring. Now, before that becomes too neat, the animal complicates it. Because krill are not the specialists this description implies. They also scrape. The tips of some of those limbs carry stiffer, rake-like bristles, and they use them on the underside of sea ice, working algae loose from a surface rather than straining it from open water. That is a completely different feeding action performed by the same set of limbs, and it becomes central to the story later. They also feed on the seafloor, in places well below the depths where anyone expected to find them. They have been observed near the bottom, in deep water, apparently working the sediment. And they are not vegetarians. Krill take copepods and other small animals when they can, which means the basket is not purely a plant filter, but a general-purpose capture device that can be turned to whatever is available. That flexibility is why they dominate. A pure filter feeder locked to one particle size would have been eliminated by the first bad summer. This animal can switch modes. There is a limit, and it is worth being honest about it. Flexible is not the same as unconstrained. Every one of those alternatives depends on something else being present. Ice with algae on it, a seafloor within reach, small animals in the water. Take away the options, and the flexibility is theoretical. One more thing about the basket, and it is easy to miss. Krill are crustaceans, which means the entire filter is part of the exoskeleton. Bristles, side branches, the whole braided mesh. All of it is external, and all of it wears down, which means the only way to maintain the most important structure the animal owns is to shed it and grow a new one. They molt constantly, through their lives, on a cycle measured in weeks rather than years, again and again, each time abandoning a complete transparent copy of themselves and building a fresh one. That is expensive. It burns energy and material every cycle just to keep the tool sharp. It also means that during the molt itself, the animal is soft, vulnerable, and briefly unable to feed properly. Hold on to that habit. It seems like housekeeping. It turns out to be one of the two ways this animal reaches down and touches the deep ocean. And one of the strangest survival tricks in the sea runs directly through it. For now, stay with the body. Because the same feature that lets you see that green gut stripe from outside, the transparency, the near invisible shell, the almost there quality of the whole animal, reads as perfect camouflage. It is not. There is one direction from which a transparent body is still a shape, still an outline, still a target. And the animals hunting krill figured that out long before we did. Transparency is a good trick and a partial one. Hold a krill up in a lab, and you can read print through parts of it. Out in the water column that ought to be the end of the story. No shadow, no outline, nothing for a predator to lock onto. Except transparency has a specific weakness, and the animal cannot fix it because the weakness is caused by the only two organs it cannot afford to make invisible. An eye works by absorbing light. That is the entire principle. Photons have to be caught and converted, which means the retina must be pigmented, which means it must be dark. Those large black compound eyes on their short stalks are the most conspicuous part of the animal, and there is no version of a functioning eye that is see-through. The gut has the same problem. A krill that has been feeding carries a bright green stripe down its length. Concentrated algae, exactly the material the whole body exists to gather. The better it eats, the more visible it becomes. So the animal is not invisible. It is a pair of black dots and a green line suspended in clear tissue. Now think about where the danger comes from. In open water, there is no floor, no wall, no cover. Threats arrive from any direction, but they do not arrive equally from all of them. Because light in the ocean has a strong preference. It comes from above. Sunlight enters the surface and weakens with depth, so at any given point in the water column, there is a bright ceiling and a dark floor. That asymmetry decides how hunting works down here. Look downward and you are looking into darkness, at animals lit only by whatever faint light has filtered past them. Look upward and you are looking at a glowing background. And anything between you and that glow becomes a silhouette. Not a color, not a texture, a shape cut out of the light. Silhouettes do not care how clear your tissue is. 2 black eyes and a full gut are enough. And they especially do not care at the depths where krill spend their days, where the remaining light is a dim blue-gray wash that turns everything above you into a shadow theater. Predators exploit this. Many midwater fish have eyes angled upward for exactly this reason, built to scan the bright ceiling for interruptions in it. Penguins and seals hunting from the surface come down and hunt back up through the light. The direction of attack is not random. It is dictated by physics. An animal in this situation has 2 options: get out of the lit zone entirely, or stop being a silhouette. Krill do both. The second one is where this gets strange. Turn a krill over again. The same underside that carries the feeding basket. And there are structures there that have nothing to do with eating. Small, rounded, embedded organs. There are 10 of them. A pair on the eye stalks, a pair at the base of 2 of the thoracic limbs, and 4 spaced along the underside of the abdomen. They produce light. Not a reflective sheen, not a chemical smear. These are constructed light organs. And what is inside them is the surprising part. Each contains a light-producing region backed by a reflector and fronted by a lens. There is a structure in there for aiming the output. Several of them can be rotated by the animal. That is a serious piece of hardware for a creature the length of your little finger. This is not a glow-in-the-dark novelty. It is a directional lamp with a reflector, a lens, and a mount. The light is blue-green. in the band that travels furthest through seawater. Photographs of it show pinpricks in black water arranged along the belly of the animal. Crowds of them in a swarm can turn a patch of ocean faintly luminous. Now ask the obvious question. Why would an animal trying not to be seen switch on lights? The intuitive answer is communication, glowing to find each other in the dark, or to attract a mate, or to signal alarm. That answer is reasonable, and it may be part of what is going on. It is also the wrong first guess, because it ignores where the organs are. They are on the underside, facing down, aimed at whatever is below. There is an established solution in the ocean that fits this arrangement exactly, and it is used by a range of mid-water animals, from certain squid to a number of deep-living fish. It is called counter-illumination, and it inverts what camouflage means. You do not hide by going dark. You hide by matching the background. And your background, when viewed from below, is glowing. So you produce your own light on your underside, at roughly the brightness and color of the faint daylight leaking down around you, and you erase your own shadow. A predator scanning upward for a dark interruption in the ceiling finds no interruption. The outline dissolves. Nothing there. Camouflage by light, not by darkness. The mechanism is well established in other animals. In krill, the case is strong but worth stating carefully. The anatomy fits it, the organ placement points downward, the emission color is right, and luminescence has been observed to follow daily and light-related rhythms. Whether counter-illumination is the primary function, one of several, or works alongside signaling, is still argued. This is the honest position. The equipment is unmistakably built for something precise, and the leading explanation for what is the shadow problem. Take the leading explanation seriously, and it produces a demand that reaches far beyond this animal. Counter-illumination is a matching problem. To erase your silhouette, you have to emit light that resembles the light behind you, the wrong brightness, and you become a beacon instead of a blank. Too dim, and you are still a shadow. Too bright, and you are now the most obvious thing in the water. But the background is not fixed. It changes with depth, constantly, because that is what light does in water. It changes with the time of day, with cloud, with the ice overhead, with the season. The value you need to match is moving, which means the animal has to know something about where it is in the light field and adjust. There is evidence that krill luminescence responds to ambient light conditions, which is what you would expect from a system doing this job. And it means depth becomes a decision with a cost attached. There is a band in the water column at any given hour where the light is dim enough that your own emission can plausibly match it and where the shadow problem is manageable. Go shallower, and the background gets too bright to compete with. You become a hard silhouette against strong light. Go deeper, and you are safer from above, but you have left the surface layer where the algae grow. That is the trap. The food is at the top. The light is at the top. The predators hunt using that light. So the place where a krill can eat is the place where a krill is most visible. And the place where it is hardest to see is the place where there is nothing to eat. You cannot solve that by staying put. There is no depth that is both safe and productive. Notice what has happened here. A small set of light organs on the belly of a shrimp-sized animal has just turned into a constraint on where the entire population can be hour by hour for its whole life. There is a complication, and it is a real one, because the Antarctic does not behave like the rest of the ocean. In high summer down there, the sun barely sets. The bright ceiling never goes away. In deep winter, it barely rises, and the whole light-based system runs differently. The shadow problem weakens, but so does everything that depends on sunlight. The neat picture of dark nights and bright days breaks down at the poles, and how krill manage that is one of the genuinely open questions about them. They appear to keep internal rhythms running even when the external light stops giving clear signals, but the details are still being worked out. Which leaves us here. Being transparent is not enough. Being small is not enough. Even carrying a set of lensed, aimable light organs on your belly is not enough, because the whole system only works within a narrow band of depth and light. And that band moves. So the animal moves with it. All of them. Every day. And when you multiply that decision by an entire population, something happens that stops being about camouflage at all. Something you can watch on a ship's sonar, as an entire layer of the ocean lifts hundreds of meters toward the surface, then falls back before dawn, on a schedule, forever. Watch a ship's echogram over a full 24 hours, and you stop seeing a picture of the ocean. You start seeing a machine? The display draws depth downward and time sideways, so the record builds as a scrolling wall. Through the middle of the day, there is a band sitting deep, a dark smear of returned sound, holding position a few hundred meters down, steady as a shelf. Then, as the surface light begins to fail, the shelf lifts. Not in pieces. The whole thing rises, hour by hour, drawn upward across the screen in a curve so smooth it looks drafted rather than lived. It reaches the top. It stays. And before the light comes back, it falls again, sinking back into the dark on the same schedule it climbed. That trace is not one swarm. It is a layer. Sonar operators encounter versions of it across the world's oceans. And when you account for how much of the sea does this and how many animals are involved, you are looking at the largest recurring movement of living material on the planet. Not the great whale migrations, not the bird routes that cross hemispheres. This one, vertical, unglamorous, happening every single night in most of the ocean at once, and almost never filmed because the animals doing it are small and the water is dark. Antarctic krill are one of the heavyweight participants, and this is where the story stops being about one animal's body and starts being about the planet. Start with the wrong explanation, because it is the one that sounds right. They go up at night to feed. The algae are near the surface, so the animals come to get them. Migration as commuting to work? That is half true, and it explains nothing, because it does not answer the actual question. If the food is at the top, why leave? Why spend the daylight hours several hundred meters below the only place your food grows? Nothing is forcing you down there. The surface layer does not become inedible at dawn. Turn it around, and the logic snaps into place. They are not going up to feed. They are going down to hide, and the feeding is what they do in the window when hiding is not necessary. Everything from the last stretch of this story applies here. During daylight, the surface layer is a lit stage. A krill up there is a silhouette against a bright ceiling, and every predator that hunts by looking upward has an easy target. The light organs on its belly can only do so much when the background overhead is blazing. So the animal retreats to depth, where the light is weak, where the shadow problem shrinks, and where it can sit out the dangerous hours. Then the sun goes down and the arithmetic reverses. The ceiling dims. The silhouette disappears on its own, and the surface becomes for a few hours a place where you can eat without advertising your position. That reframes the whole thing. This is not a commute. It is a scheduled trade, run daily, between two things that cannot both be had at once, food and safety. And the price is brutal. Remember the fundamental condition of this animal. It has no float, no gas, no buoyancy aid. It sinks the moment it stops beating those paddles. Now ask it to climb. Rising several hundred meters means swimming upward, actively, against its own weight, through cold water for hours, then reversing the whole thing before dawn, every day, for years, not as an occasional migration event with a rest at the other end, but as the normal daily rhythm of being alive. The energy that goes into that has to come from somewhere, and it comes out of the same budget the animal is trying to fill in the few hours it spends feeding, which means the trip has to earn back more than it costs, or the strategy collapses. That it persists across the species, across the ocean, across the year, tells you how heavy the predation pressure at the surface must be. The animals are paying an enormous, permanent energy tax to avoid being seen in daylight, and the tax is still cheaper than the alternative. Nothing pays that much unless the alternative is worse. And the tax is not paid once. It compounds. An animal that spends part of every night climbing has less left over for growth, for building reserves, and for producing eggs, which is a cost the population ends up paying in a later season, in a different currency. Now the picture at the surface, because it is worth seeing. A ship working at night with deck lights on can find the water alongside boiling. Krill at the surface in numbers close enough to scoop, the water thick with them, where hours earlier there was nothing. Seabirds work the same shift. So do some of the seals. The whole surface ecosystem at night is running on animals that were not there at noon and will be gone by morning. Then, before first light, it drains away. Here is the part that starts to matter beyond biology. The animals go up empty and come down full. They feed at the surface. Guts loaded with green, packed with algae, they have strained out of the top layer. Then they descend, carrying that material down with them into water hundreds of meters deep. And while they are down there, they process it, which means digestion happens at depth. And what comes out of a krill comes out down there, in the dark, far below the surface, where the carbon was originally captured. Stop and look at what has just been described. Carbon dioxide dissolves into surface water. Algae take it up and build themselves out of it. Krill filter those algae and swallow them at the top of the ocean, then physically transport that material downward in their own bodies and release it at depth. That is not an animal moving. That is carbon moving in a specific direction, driven by the daily rhythm of an entire population. The migration is a pump. It runs on nothing but the fear of being seen, and it moves material out of contact with the atmosphere, downward, every night, across an ocean that wraps the planet. Before this gets too clean, the animal complicates it again, and honesty requires saying so. Krill do not all do the same thing. Some swarms stay near the surface for extended periods. Some feed at depth. Reverse patterns have been recorded, animals ascending during the day. Behavior varies with season, region, food availability, ice cover, the presence of predators, and the age of the animals. Larvae and adults do not always follow the same schedule. And there is the polar problem. Near the pole in high summer, the sun does not set. The light cue that organizes this rhythm across most of the ocean weakens or vanishes. In deep winter, the reverse. Krill appear to keep some internal timing running through those stretches, but how they manage the transition and what the migration looks like under continuous daylight is genuinely unsettled. So the clean image of a single layer rising and falling on a strict clock is a summary, not a law. The pattern is real and it dominates. It is not universal. None of that weakens the point. It makes it larger, because it means the pump is not a fixed mechanism, but a behavior that responds to conditions. Which means it can change when conditions do. There is one more layer, and it goes deeper than the daily rhythm. Beyond this nightly rise and fall, there is a seasonal version. Krill have been recorded much deeper than the migration layer, down near the seafloor in water far below the range surveys usually cover. And there is growing interest in what fraction of the population makes those deeper excursions and why. Feeding on the bottom is one possibility. Avoiding predators is another. The extent of it is an open question. And it is one of the reasons the counting problem from earlier remains as stubborn as it does. Whatever the answer, the direction of travel is the same. Material captured at the top ends up somewhere well below. So step back and look at what this animal has become over the last few minutes. It began as a translucent thing on a fingertip, with a filter for legs and lamps on its belly. Now it is one unit in a population-scale conveyor, lifting and dropping through hundreds of meters of water on a daily cycle, dragging the products of surface sunlight down into the cold and leaving them there. That is a much bigger job than being food. And it raises a question that changes the scale of this entire story. Because the animals come back up the next night. Whatever they left behind does not. What goes down stays down. And what a krill leaves in the dark turns out to be the single most consequential thing it produces. Not its body, not its meat, the material it discards. Point a light down into black water and wait. Give it a minute and the beam stops looking empty. Small pale flecks begin drifting through it, all moving the same way, all moving down. Some are ragged and slow. Others are compact and quick, dropping through the cone of light and gone before you can focus on them. Divers and submersible crews have a name for it: marine snow. It falls constantly, everywhere in the ocean, at every hour, and it has been falling for as long as there has been life in the surface layer. Most of it is junk. Fragments of dead cells, mucus, broken bits of organism clumped together into loose flakes with the density of wet tissue paper. That kind of particle sinks slowly, tens of meters a day, sometimes less. It drifts for weeks in the upper water, and while it drifts, bacteria work on it, breaking it apart, releasing the carbon inside it back into the water where it can dissolve, circulate, and return to the surface. That is the default fate of almost everything that dies in the ocean. Down a little, consumed, recycled. Nothing leaves. Now watch the fast ones. Among the drifting flakes, there are particles that behave completely differently. Compact, dark, cylindrical, dense, and dropping through the beam at a speed the others never approach. They do not tumble. They do not disintegrate. They fall like something that was built to fall. Those are krill fecal pellets, and they are the reason this animal matters to the planet, rather than just to the southern ocean food web. Here is why they fall so fast. A krill packs its waste into a tight cylinder wrapped in a membrane, and the contents are not soft. Remember what these animals are eating in a productive Antarctic summer: diatoms, the large armored phytoplankton, whose cell walls are made of silica, glass essentially. So the pellet leaving a krill is a compressed tube of mineral shell fragments and organic material held together with a density far higher than a loose flake of mucus. Dense and compact means fast. These pellets can sink at rates measured in hundreds of meters per day rather than tens. Some fall faster still. Change the speed and you change the destination. This is the entire point, and it is worth being precise about it. A carbon particle drifting slowly through the upper ocean is not removed from anything. It sits in water that will contact the atmosphere again within months or a couple of years. Bacteria strip it, and the carbon goes right back into circulation. From the atmosphere's perspective, nothing happened. A particle that sinks fast enough to get below a certain depth is in a different world. Deep water does not return to the surface quickly. Depending on where it goes and which current it joins, the water down there can stay out of contact with the air for decades, centuries, or longer. Carbon that arrives down there is not gone forever, but it is out of the conversation for a very long time. So the question is never whether carbon sinks. It is how deep it gets before something eats it. Speed decides that. A slow particle is intercepted. A fast particle escapes. There is a rough threshold involved, and it is one of the more important lines in ocean science that nobody can draw precisely. Somewhere below the sunlit layer, past the depth where most consumption happens, a sinking particle crosses from being temporarily out of sight to being genuinely removed. Where that line sits depends on the region, the season, and the circulation underneath it. What is not in dispute is that the line exists, and that almost everything sinking through the ocean fails to cross it. Krill build fast particles, and they build them in a place that makes the geometry even better. Go back to the migration. The animals feed at the surface at night, then carry the full gut down hundreds of meters before dawn. Digestion happens down there, so the pellet is not released at the surface, with a kilometer of hungry water beneath it. It is released already deep, in the dark, with much of the dangerous zone already skipped. The animal has done the first leg of the journey itself, using muscle. Then there is the second thing they drop, and it is easy to overlook because it does not look like waste at all. Remember the molting, that entire feeding basket, the braided mesh, the branching bristles, the shell, the eye coverings, is exoskeleton, and it wears out, so krill shed the whole thing and grow a new one, over and over, on a cycle of weeks. What they leave behind is a complete transparent copy of the animal, perfect, hollow, empty. In water with a lot of krill in it, these things drift in numbers, ghost krill turning slowly in the current. They also sink, and they take material with them, including the chitin and minerals that made up the shell, plus whatever organic residue clung to it. Stack the two together, and you get something odd. This animal exports carbon downward, through what it eats, what it excretes, and what it discards. Its body being eaten by a whale is only one of several pathways, and arguably not the most important one for the deep ocean. The most consequential thing a krill produces is garbage. Now the honest caveat, because there is a real one, and the science here is active rather than settled. Not every pellet makes it. Fecal pellets are food. Other animals in the water column eat them, including other krill, and bacteria attack them on the way down. A pellet that gets intercepted at 200 meters has not exported anything meaningful. Pellets can also break up, and fragments sink slower than whole ones. There is also a behavior that complicates the picture. Krill sometimes feed at very high rates when food is abundant and pass material through fast, releasing pellets that are still rich in usable content. Efficient for the animal in a bloom, but it makes the pellet more attractive to whatever finds it on the way down. So the fraction of what leaves a krill that reaches genuine depth varies. With region, season, food supply, water conditions, and what else is living in the intervening water. Researchers work on this using sediment traps, which are essentially funnels moored in the deep collecting what falls, and the numbers that come out are measurements of specific places at specific times, not a global constant. What is not in doubt is the mechanism, and the mechanism is the story. Krill-produced particles are among the faster sinking material in the ocean, and they are produced in enormous quantity across a vast region. Any serious accounting of how carbon moves out of the Southern Ocean surface has to include them. Be careful about the language. This is not a claim that krill are quietly fixing the climate, and it is not a claim that anyone can put a confident global tonnage on. Efforts to quantify it exist, and the estimates vary widely, which is exactly what you would expect for a process this patchy operating across an ocean this large. Where a number is uncertain, the mechanism is what should be trusted. So picture what the water column actually is. Not a body of water with animals in it. A chute. Sunlight lands at the top and gets captured by cells too small to see. Those cells get strained out and concentrated by an animal the length of your finger. That animal carries the material down through several hundred meters of water using its own muscle, then packs what it cannot use into dense cylinders and lets go. Then it swims back up and does it again the next night. A biological elevator, running in one direction, powered by fear of predators, moving carbon from the atmosphere's doorstep into water that will not see the sky again for a very long time. And here is what makes it strange rather than merely impressive. Everything just described removes material from the surface. Carbon down, nutrients down, silica down. That is a system steadily impoverishing the layer it depends on, because the algae at the top need more than sunlight and carbon dioxide to grow, which sets up a problem the Southern Ocean has never fully solved. There is a nutrient down there that is desperately scarce, so scarce that vast stretches of that ocean sit full of everything else a plant could want and still stay barren. Water thick with nitrate and phosphate, bathed in summer sunlight, and almost nothing growing in it. Something is missing. It is present in quantities so small it is measured in traces, and it turns out that a meaningful amount of it in the Southern Ocean is not dissolved in the water at all. It is inside the krill. Put a satellite map of ocean productivity on the screen and let it run through a Southern Hemisphere summer. The color scale shows where photosynthesis is happening. Blue is barren. Green and yellow mean cells are dividing. Around the Antarctic coast, along the ice edge downstream of islands, the map lights up hard, bright ribbons of growth curling out into the sea. Then look at the water between those ribbons. Enormous stretches of the Southern Ocean stay blue through the entire summer, not just dim. Persistently, stubbornly blue, year after year, in water that should be exploding. That water is not poor. That is the confusing part. The Southern Ocean is one of the most nutrient-rich surface environments on the planet. Deep water rises there, carrying nitrate and phosphate up from the ocean interior in concentrations that would drive a bloom almost anywhere else on Earth. The sunlight arrives in summer, continuously, for weeks. The ingredients are laid out, and nothing happens. Oceanographers have a name for this condition, and it appears in a few regions around the world. High nutrient, low chlorophyll. Plenty of food, no growth. For a long time, it was one of the more embarrassing problems in ocean science because the obvious explanation kept failing. Grazing pressure alone did not account for it. Light did not account for it. Temperature did not account for it. The answer turned out to be a shortage so small, it had been below the reach of the instruments used to look for it. Iron. Every photosynthetic cell needs it. It sits inside the machinery that runs photosynthesis and the machinery that converts nitrate into usable form. Without iron, a cell cannot build the equipment to use the nitrate sitting all around it. The nutrients are there. The tools to process them are not. The amounts required are minuscule. Trace quantities, orders of magnitude below the other nutrients, which is exactly why it took so long to identify. Early attempts to measure iron in seawater were contaminated by the ships and equipment doing the measuring. Steel hulls, wire cables, metal instruments. Researchers were finding iron because they had brought it with them. Getting a clean number required rebuilding the whole sampling process to keep metal away from the water. Once that was solved, the map made sense. The blue regions are not starving for food. They are missing a trace metal. There was a way to test it directly, and it is an unusually blunt one. If iron is the limit, then adding iron to the surface of a high nutrient, low chlorophyll region should produce growth where none was happening. That has been done at sea, deliberately, in a series of ocean experiments including work in Southern Ocean water. And the result was a bloom. Algae responding to a trace metal dropped into water that already had everything else it needed. That is about as clean a demonstration as this field gets. It also opened an argument that has never really closed, about whether deliberately fertilizing the ocean is a sensible thing to attempt, and what happens to the carbon afterwards. Set that argument aside. The relevant part here is what the experiments established. In this water, iron is the switch. Now the geography, because it explains the pattern on the satellite image. Iron gets into surface seawater from a handful of sources: dust blown off continents, sediment stirred up around coastlines and shallow shelves, meltwater and material carried by ice, deep water rising in specific places. Look at where those are, and look at the bright ribbons again. They match. Growth clusters near coasts, over shallow ground, along the ice edge, downstream of islands. All the places where iron gets delivered. The open ocean far from land gets almost none and stays blue. The Southern Ocean is a field with fertilizer spread only at the edges, which raises the question that makes this chapter matter. Iron does not stay in the surface layer by itself. It gets used, incorporated into cells, and then those cells die or get eaten and the material sinks. Everything described a moment ago. The fast pellets, the molts, the biological elevator running downward, takes iron with it, out of the sunlit layer, into deep water where no plant can reach it. So the system has a leak. Every day, the pump that moves carbon down is also moving the scarcest resource in that ocean down. Given enough time, the surface should strip itself bare. Something has to be putting iron back. This is where krill stop looking like a food item and start looking like part of the plumbing. Krill accumulate iron in their bodies. They eat algae that have concentrated it, and the metal ends up in their tissue at levels far above what is dissolved in the water around them. Multiply that by the biomass involved, and the Southern Ocean's krill population represents a substantial pool of iron, not dissolved in the sea but locked inside animals, held in the upper water column rather than sitting on the seafloor. And that pool does not stay sealed. Krill excrete. They molt, shedding shell material into the water. They get torn open by predators. They die and decompose. Every one of those roots releases iron back into the water column, and much of it happens in the surface layer where it can be used again. The animals are not just moving material downward. They are also holding a share of it up and leaking it back into the light. Then there is the pathway that got the most attention, and it deserves both credit and caution. Whales eat krill in enormous quantities and concentrate everything those krill contain. What comes out of a whale is released into surface water in large, buoyant plumes, and analyses have found iron in whale feasts at concentrations far above the surrounding seawater. The idea that whales help fertilize the very system that feeds them, that they take a resource locked in animal tissue and return it, dissolved, to the sunlit layer where algae can use it, has become one of the more compelling ideas in Southern Ocean ecology. It is also still an area of active work rather than a closed case. How much of that iron is in a form algae can actually take up, how far the plumes spread, and how much total productivity depends on this pathway compared with dust and sediment sources. These are open questions. The mechanism is documented. The magnitude is argued about, but even the cautious version of this changes how you have to think about the food web. The standard picture is a ladder. Algae at the bottom, krill above them, whales at the top, energy flowing upward, each step consuming the one below. Predators take, prey give. Simple. That picture is wrong, or at least badly incomplete, because if predation returns iron to the surface, then a whale-eating krill is not only removing krill, it is also releasing, in usable form, the one thing that limits how much food the entire system can grow in the first place. The consumer is feeding the producer. That is not a ladder. It is a loop. Material cycles around, rather than flowing one way, and every large animal in the Southern Ocean sits inside that circuit rather than on top of it. And krill are the point where the loop concentrates. They gather diffuse iron from the water into animal tissue. They hold it in the surface layer while they live. They release it through excretion and molting, and when something eats them, they deliver a concentrated package to an animal that will return a large fraction of it to the light. Take the krill out, and you have not removed a link. You have opened the circuit. The iron does not vanish. It sinks, because without animals catching it and holding it up, the natural direction for a particle in seawater is down. The surface layer gets poorer. Poorer surface water grows less algae. Less algae means less material available to the animals that remain. That is why this cannot be treated as subtraction. Remove a predator from a normal food chain and the prey population rises. Remove a species that participates in nutrient recycling, and the effect runs in directions that a simple chain does not predict. Which sets up the most uncomfortable question in this entire story. In the 20th century, industrial whaling removed an enormous share of the great whales from the Southern Ocean, millions of animals, the largest krill predators on Earth, taken out of the system in a matter of decades. If the latter model were right, the outcome would be obvious. Remove the predators, and the prey should have exploded. Krill everywhere. The Southern Ocean thick with them, more than anyone had ever seen. Scientists at the time expected exactly that. It was a reasonable prediction, and it had a name. The surplus never clearly showed up. The numbers involved are not subtle. Across the 20th century, industrial whaling in the Southern Ocean killed great whales on a scale that has no equivalent in the history of hunting. Blue whales, fin whales, humpbacks, minkies. Populations reduced by percentages that in some cases ran into the high 90s. Whole species pushed to a few percent of what they had been. Every one of those animals was a krill predator, the largest one that has ever existed, eating krill by the ton, day after day, through every feeding season. Take that out and the consequence should be arithmetic. Fewer mouths means more prey. That was the expectation, and it was not a careless one. It was a reasoned prediction from people who understood the system as well as anyone then could. And it had a name. The krill surplus hypothesis. Remove millions of enormous consumers, and the animals they consumed should be left with a windfall. Krill should have surged. Other krill eaters, seals, penguins, seabirds, should have expanded into the space the whales left behind. The prediction was clean, testable, and reasonable. It has not held up cleanly. Be careful here, because this is exactly the kind of place where a documentary starts overclaiming. Some parts of the prediction did find support. There is evidence that certain krill predators increased during that period, and various lines of argument have been made about penguin and seal population changes in the decades after whaling. Regional stories differ. The picture is not uniform, but the central expectation: a Southern Ocean visibly loaded with surplus krill, an obvious overflow, a system with more of this animal than it knew what to do with, has never been demonstrated in the way the hypothesis implied. And the long-term work on krill abundance in some regions has pointed in the opposite direction, toward decline in parts of their range over recent decades. That is a strange result. It deserves to be treated as strange. There are several candidate explanations, and honesty requires laying them out rather than jumping to the most dramatic one. Start with the boring possibility, which is also the most important. The measurements might not be good enough to answer the question. Everything from earlier applies. Krill are patchy, they dodge nets, they reflect sound unpredictably, they swing wildly between years, and systematic surveys only began well after whaling was already in full swing. There is no reliable measurement of Southern Ocean krill abundance from before the whales were removed. The baseline everyone would need to detect a surplus does not exist, so one legitimate answer is that a surplus may have occurred and simply passed unrecorded. In an ocean nobody was measuring properly, decades before anyone had the tools. The second explanation is climate. The 20th century was not a stable background against which a single experiment ran. Sea ice conditions changed, water temperatures changed, regional oceanography changed. Any signal from whaling is tangled with those shifts, and separating them across a century of patchy data is genuinely difficult. There is also the question of who moved in. Taking the whales out did not leave krill alone in the water. Other predators were already there, and some of them respond fast to an opening. Seals and penguins breed on land and can expand quickly when food allows. If those populations grew into the gap, predation pressure on krill would not have vanished so much as changed hands, which would flatten any surplus before anyone could measure it. That case has been argued in both directions, and it runs into the same wall as everything else here. The records that would settle it were not being kept in the places and during the years that would have mattered. The third possibility is the one that reorganizes everything, and it comes straight out of the iron problem. Whales were not only consuming krill, they were also processing them, taking iron locked in krill tissue and returning a share of it in usable form to the sunlit surface layer where algae grow. Follow that logic, and the ladder inverts. The whales were a component in the machinery that kept the surface layer fertile. They intercepted material that would otherwise sink, concentrated it, and released it back into the light. Under that view, removing the whales did not just lift predation pressure. It removed a substantial recycling pathway from the top of the system. Less recycling means less iron staying in the surface. Less iron means less algal growth. Less growth means less food for krill, which produces a conclusion that sounds broken until you see the mechanism. Killing the predators may have reduced the prey, not through anything the whales did to the krill, through what the whales did for the water. Sit with how much that overturns. Every intuition about ecosystems runs on the idea that predators suppress prey. That is the model everyone carries. The wolf and the deer, the lion and the herd. Take the predator away and the prey rises. It is the most basic relationship in ecology. Here, the relationship may run partly the other way. The consumer may have been sustaining the conditions its own food depended on. Predation as maintenance rather than pressure. Now hold that at arm's length because it is a hypothesis and not a settled result. The whale pump idea is grounded in real measurements of iron in whale feces and in solid reasoning about how nutrients cycle. What is not established is the magnitude. Whether whale recycling was ever large enough to be a controlling influence on Southern Ocean productivity as a whole, or whether it was a real but secondary contribution alongside dust, sediment, upwelling, and the recycling done by krill and everything else. Researchers have estimated it in various ways. The estimates do not agree, and the honest position is that the mechanism is documented and the scale is argued. There is a fourth explanation, and it may be the most likely of all. Several of these are true at once. The measurements are inadequate. The climate shifted. The recycling loop was damaged. Regional patterns differ. A commercial krill fishery arrived, all of it overlapping across the same decades, in an ocean too large to survey properly, with no clean baseline to compare against. That is not an unsatisfying answer. That is what a real system looks like when you try to reason about it after the fact. And the awkward part is that the experiment cannot be run again. There is no second Southern Ocean sitting untouched alongside this one, available as a control. And the deeper lesson from this stretch of the story is not about whales at all. It is about what happens when a food web is treated as a hierarchy. The old model was not stupid. It was the standard framework, applied competently to the largest predator removal in recorded history. What it missed was not a fact, it was a direction. Everyone was tracking energy moving upward, from algae to krill to whale, because that is the direction food moves. Nobody was tracking material moving back down and sideways, because for most of that century, nobody knew iron was the limiting factor, and the tools to measure it did not exist. You cannot include a mechanism you have not discovered yet. So science updated itself, and the updated version is harder to work with. The clean prediction is gone. What replaced it is a system where removing a species can produce effects that run in unexpected directions, and where the largest ecological experiment ever conducted was performed without a control, without a baseline, and without anyone measuring the variable that turned out to matter, Which brings the story to the practical point. If the whale recycling loop was significant, then a partial recovery is underway. Several great whale populations in the Southern Ocean have been increasing since commercial whaling ended, some substantially. That should mean more krill consumption. It should also mean more recycling. Which of those effects dominates, and what it does to krill numbers, is being watched right now. An ongoing natural experiment nobody designed, running in real time, in an ocean we still cannot count. And nobody is going to get a clean answer out of it either, for exactly the reasons that made the original question so hard to settle. But notice what has been missing from every explanation so far. Whaling, iron, recycling, predation. All of it concerns the top of the system. Large animals doing large things to the water. Meanwhile, the thing that most reliably determines whether a generation of krill survives is not a whale, and not a fishery, and not a nutrient. It is a physical structure that forms and disappears every year, that covers millions of square kilometers and then retreats, and that most people assume is nothing but a frozen lid over dead water. It is the ice. And the assumption that it is a barrier is the exact opposite of what it is. Everyone has the same mental image of Antarctic sea ice, and it is wrong in a way that matters. The image is a lid, a white sheet closing over the ocean, sealing it, blocking the light, ending the growing season. Under it, cold, dark water, waiting for spring. A barrier between a living surface and a dormant sea. Get a diver under it, and the picture inverts immediately. The underside of Antarctic sea ice is not white. In the right season, it is stained brown and gold. Patches, streaks, whole sheets of discoloration running across the ceiling, dense enough to shade the water below. From beneath looking up, it can resemble rust spreading across a frozen sky. That color is life. Sea ice does not freeze into a clean, solid block. As seawater freezes, salt is excluded from the ice crystals, and it concentrates into pockets and channels running through the structure, a network of tiny brine-filled passages threading through the ice, staying liquid because the salt keeps them from freezing solid. The ice is porous. It has plumbing. And algae live in it. They colonize those brine channels and the rough undersurface, growing inside the ice and hanging off the bottom of it in strands and mats. They can tolerate cold, they can work with the dim light that filters through, and they build up in concentrations far higher than the open water around them. So the ceiling is not a barrier. It is a surface, with a crop growing on it. It is also not flat. Wind and current push ice sheets into each other, and where two of them collide, the edges buckle and break, driving blocks downward into the water. What forms underneath is not a lid, but a landscape. Ridges, keels, overhangs, hollows, loose crystal layers hanging off the base of the ice. Seen from below, it looks less like a roof than like a cave system with a light source somewhere behind it. That texture turns out to matter as much as the algae, and for a completely different reason. Now put the animal back into the picture, because this is where the whole story of Antarctic krill either works or falls apart. The problem is winter, and it is a severe one. In winter, the sun barely rises. The phytoplankton in open water, the diatoms that summer krill filter out by the millions, collapse. The bloom is over. The green stripe in the gut disappears because there is nothing to fill it with. Adults have options. That is the next part of this story. Larvae do not. A larval krill is a fraction of the size of an adult, with almost no reserves. It hatched during the previous season. It has barely begun to build a body, and it faces months of darkness in the coldest ocean on Earth with no fat to burn. By any straightforward accounting, it should not survive its first winter. What it does instead is go to the ceiling. Under ice observations have found larval krill concentrated on the underside of sea ice, in the rough zones, along the ridges, in the crevices and overhangs where the structure has been broken and refrozen. And they are not sheltering there passively. They are grazing. Remember the feeding basket? And the detail from earlier that looked like a minor variation. The stiffer, rake-tipped limbs sitting among all that fine mesh. In open water, the basket strains particles out of a current. On ice, those rakes work like a scraper, prizing algae off a solid surface and pulling it back toward the mouth. The same equipment, used two completely different ways in two completely different environments, depending on the season. So a larval krill spends the polar night hanging beneath a frozen ceiling, upside down, scraping food off it. That behavior is documented, and it does not resemble what most people imagine plankton doing. The ice is a pasture, grazed from below, and it does two jobs at once, which is why it matters so much. It provides winter food where there is none in open water. It also provides physical structure. The ridges and cavities on the underside offer places to hide in an environment where the water column has no cover at all. For a defenseless animal in a world of open water, three-dimensional structure is rare and valuable. Food and cover, in the one season when both are otherwise unavailable. There is a third benefit, and it arrives at the far end of the winter. When the ice melts back in spring, it releases the algae it has been holding, and it lays a layer of fresher, lighter water across the surface that helps keep cells suspended in the light. The result is a bloom that follows the retreating ice edge across the ocean like a slow wave. The animals that spent the winter underneath the ice are already in position when it starts. That combination explains something that had been sitting unaddressed since the start of this story: why krill are where they are. The distribution of Antarctic krill is not even. They concentrate heavily in particular regions, and those regions correspond to areas with reliable seasonal ice and the right circulation. The animal is not distributed by chance across a uniform ocean. It is pinned to a physical feature that forms, retreats, and reforms every year. Now, the part that turns this from natural history into a problem. That feature is seasonal, variable, and not guaranteed. Sea ice extent in the Antarctic swings enormously through the year, a vast area freezing and then melting back every 12 months. It also varies between years, sometimes dramatically, and the regional patterns differ. Some areas have shown different trends from others. Antarctic sea ice behavior has been more complicated than the Arctic's and has produced genuine surprises, including recent years with extents well below the long-term range. Set aside the projections because they are not needed to make the point. The mechanism alone is enough. If larval krill depend on the ice for winter survival, for both food and shelter, then the quantity and timing of ice becomes a control on how many of them make it through to spring. Not a background condition, a determinant. Ice forms late and there is a gap in the winter food supply during the months when larvae have nothing else. Ice retreats early, and the season ends before the animals have built enough reserves. Ice forms thin and flat, and the ridges and cavities that provide shelter are reduced. Ice fails to reach a region at all, and that region stops functioning as a nursery. None of that requires the population to be poisoned, hunted, or starved directly. It requires only that a physical structure show up less reliably at a specific time of year. And that connects to something noted much earlier and left hanging. Krill abundance swings violently between years, with reproduction succeeding spectacularly in some seasons and failing almost entirely in others. Ice conditions are among the strongest candidates for why. Good ice years and poor ice years plausibly produce good and poor year classes, and a single strong class can carry the population for years afterward, which means the animal's numbers are tied not to a slow average, but to whether particular seasons happen to work out. There is honest uncertainty here worth stating. Exactly how much of a krill generation depends on ice algae versus other winter food sources is still being studied. Larvae have been found in areas and conditions that complicate a simple ice-only story. The relationship between ice extent, ice quality, and krill recruitment is an active field, not a solved equation, and researchers disagree about how tight the coupling is. What is not disputed is the association. Antarctic krill and Antarctic sea ice are linked in distribution, in seasonal timing, and in the biology of the youngest animals. So step back and look at what has just been established. The animal that concentrates the Southern Ocean's energy, that drives one of the largest carbon transports on Earth, that holds iron in the sunlit layer, that feeds the largest animals ever to exist. The survival of its next generation runs through a layer of frozen seawater that appears and disappears every year. The whole system has a hinge, and the hinge is made of ice. That is the nursery. But the nursery only covers the young, because the adults are out there in the same winter, in the same darkness, in the same collapsed food supply, and most of them are not clinging to the underside of the ice. They are in open water months from the next bloom, with a body that has to keep swimming every second or sink. They should starve. Their metabolism should burn straight through whatever they built up in summer, and by the time the light returns, there should be nothing left of them. Something else happens instead, and it breaks a rule so basic that most people have never thought to question it. Winter in the Southern Ocean is not a lean season. It is an absence. The light collapses. The bloom that fed everything through summer ends. Open water that was thick with diatoms goes clear and empty, and stays that way for months. There is no reduced ration to scrape by on. For an adult krill in open water, away from the ice, the food supply approaches zero and remains there through the polar night. Now recall the animal's fixed cost. It sinks if it stops swimming. There is no shutting down, no settling on the bottom to wait, no burrowing in and going still. Those paddles keep beating through the entire winter because the alternative is falling out of the water column. So the arithmetic is grim. Months of no food and a metabolic bill that never stops arriving. Every animal facing this situation uses some version of the same toolkit. Store energy in advance. Burn it slowly. Reduce activity. Krill do all three. They build lipid reserves during the summer feeding season and draw them down through the winter. They lower their metabolic rate substantially, cutting the running cost of being alive. They reduce feeding effort when there is nothing to feed on. That is standard. Every hibernating mammal, every overwintering insect, every fish in a cold season does something along those lines. Krill do one more thing, and it is not standard at all. They get smaller, not thinner, not lighter in the way a starving mammal loses fat while its skeleton stays the same size. Smaller, shorter, physically reduced in overall body length so that an animal measured in autumn and measured again at the end of winter is a smaller animal than it was. That should not be possible for something with a hard external shell. A crustacean's exoskeleton is rigid. It cannot be gradually resized, which is exactly why krill can do it, and most animals cannot. The mechanism runs through the molt. Remember that these animals shed their entire exoskeleton repeatedly on a cycle of weeks throughout their lives. The shell, the eye coverings, the feeding basket with all its grated bristles, discarded whole and rebuilt. That process is normally how a crustacean grows. Split the old shell, emerge with a soft new one, expand before it hardens, end up larger. Krill run it backwards. Under sustained food shortage, an animal can come out of a molt smaller than it went in. The new exoskeleton is built to a reduced size. Do it again at the next molt, and again, and the animal shrinks step by step through the winter. The regression is not only about length. Under prolonged starvation, adult krill can lose the external features that mark them as sexually mature. The specialized structures adults carry can regress toward a juvenile condition so that an animal that spawned last summer can, by the end of winter, resemble something that has never bred at all. an adult becoming, functionally and visibly, less adult. Then the light returns, food comes back, and they rebuild, grow again, redevelop the reproductive features, breed the following season. This is not decline. It is a reversible, repeatable strategy. And there is evidence it can happen more than once in a lifetime, in an animal that may live several years. Take a second on why this is such a good solution, because the logic is precise. Metabolic demand scales with body size. A smaller animal costs less to run. So when the food stops, instead of maintaining a large body on reserves that will run out, the krill reduces the body itself, dismantling tissue, using it, and rebuilding at a size the winter can actually support. It is not waiting out the shortage. It is resizing itself to fit the shortage. Shrinking is also not the only tool in the winter kit, and the animals appear to mix them. Some feed on whatever the dark still offers. Detritus drifting down, small animals. Material near the bottom. Some lean harder on the lipid they built in summer. Some sit deeper, where movement costs less, and predators are scarcer. Populations in different regions seem to rely on different combinations, which is part of why the polar winter is so hard to summarize and so hard to watch. What unites all of it is one principle. When you cannot increase income, you cut what the machine costs to run. And this animal can cut further than almost anything else its size. Compare it to what a mammal does. A starving mammal burns fat, then muscle, and its frame stays the same. It is a large animal running on empty. A krill in the same position becomes a small animal running on less. And small is survivable in a way that large and empty is not. Then, the cost, because there is always one. Shrinking is not free, and the price lands on reproduction. An animal that has spent the winter regressing has to spend the early part of the following season rebuilding, regrowing tissue, redeveloping reproductive structures, before it can breed. That takes time out of a summer that is already short. In a bad enough year, some animals may not get back to breeding condition in time to contribute at all. So the trade is survival now against reproduction later, which means a harsh winter does not just thin the population. It can suppress the next generation from animals that survived perfectly well. Now the consequence nobody expects, and it reaches back to a problem raised early in this story. If a krill can shrink, then body size no longer tells you age. That is a serious problem, and it undermines a technique that works across much of marine biology. For many animals, you can estimate age from size, or from growth marks laid down in hard structures, rings in a fish otolith, layers in a shell. Krill provide neither. They shed their exoskeleton constantly, so nothing accumulates a permanent record, and their length can go up or down depending on how the last winter went. A large krill might be old. It might be young and well-fed in a good year. A small krill might be young. It might be an older animal that spent the winter shrinking. Two animals of identical length can have completely different histories, and there is no reliable way to tell them apart by looking. That corrupts something practical. Population assessments often depend on understanding age structure, how many young animals are coming through, how many old ones remain, whether recruitment succeeded. With size unreliable as an age marker, researchers work with size frequency distributions, cohort tracking across seasons, and laboratory growth studies, all of which involve assumptions that shrinkage complicates. So one of the reasons the counting problem is as stubborn as it is turns out to be this trait. The animal's survival strategy interferes with our ability to assess it. The uncertainty is not only about how many, it is about who they are. A note on the limits of what is known, because this is a place where it is easy to overstate. Shrinkage in Antarctic krill has been demonstrated under controlled starvation and observed in the field, and it is accepted as a real capability. What is less settled is how widespread it is across the population in a typical winter, how much variation exists between regions and age groups, and exactly how much winter survival depends on shrinking versus lipid reserves, reduced metabolism, ice algae, or opportunistic feeding on whatever animals and detritus are available in the dark. Different studies emphasize different combinations. The toolkit is agreed on. The proportions are not. What matters is what the capability implies. This animal is not fragile. It is one of the more robust survival machines in the ocean, capable of collapsing its own body to match a bad year and rebuilding when conditions improve, an option that most animals simply do not have. Which makes what is happening to krill now harder to dismiss, not easier. If a population declines in a region, it is not because the individuals could not handle a hard winter. They are equipped for hard winters at a level that borders on the extreme. But every one of those tools is a way of surviving a gap, shrink and rebuild, burn reserves and refill, reduce metabolism and recover. All of it assumes the good season arrives on the other side. And there is one part of this life cycle where none of it helps, because the animals involved have no reserves to burn, no body to shrink, and no ability to wait: the eggs. What krill do to reproduce is one of the strangest journeys in the ocean, and it goes in a direction nobody would predict. Before a new generation can rise to the surface, it first has to fall. Hundreds of meters, into the dark, with no way back except to climb. A spawning krill releases eggs into the surface layer during the productive season, which is what almost every marine animal does. Then the eggs sink, not slowly, not accidentally. They are denser than seawater, and they drop out of the sunlit water where they were released, down through the layer where predators hunt, down past the depths where the daily migration turns around, continuing for hundreds of meters and in some places well beyond 1,000. They keep falling for days, and while they fall, they develop. The embryo divides and organizes in a container that is descending the entire time, so that by the time hatching occurs, the animal is nowhere near where it started. It is deep, in cold water, in the dark, at a depth where nothing it needs exists. Then the newly hatched larva has to climb all the way back. This is called the developmental ascent, and it is one of the more demanding things a small animal does anywhere in the ocean. The larva that hatches down there is not a functional feeder. It has yolk and only yolk. It cannot eat. The feeding equipment is not ready, and the food is not there anyway because algae need light, and there is none at that depth. So it swims up, hundreds of meters, through cold water on a fixed and non-renewable fuel supply, with the requirement that it reach the sunlit layer and complete its development before the yolk runs out. There is no second attempt. There is no pausing to recover. It is a single climb on a fuel tank that was filled once against a deadline set by its own metabolism, and the body changes shape while the climb is happening. Krill do not hatch as small krill. They hatch as something that barely resembles the adult, with a fraction of the equipment, and they pass through a run of distinct larval stages on the way up and after they arrive, molting between each one, rebuilding themselves progressively as they go. The feeding basket that defines the adult animal is not there at the start. It has to be assembled, stage by stage, and it has to be finished before the yolk is gone. So this is not only a journey, it is a construction project running inside a body that is falling behind on fuel the entire time. That is the gauntlet. Now the obvious question, why on earth would a species do this? The straightforward answer is that the surface is lethal for something that small and helpless. Everything about the upper layer that makes it productive also makes it dangerous. It is where the food is, which means it is where the feeders are. A krill egg or a newly hatched larva sitting in that water has no speed, no defense, no camouflage worth mentioning, and no ability to escape. It is protein in a well-attended dining room. Depth removes that problem. Not by hiding. Depth is genuinely a different environment. It is dark. It is cold. And the density of animals capable of finding and eating a drifting egg is far lower. Sending the most defenseless stage of the life cycle down into that emptiness for its most vulnerable period is a real solution, and the cost is the climb. There may be a second advantage in the descent itself. The deep water in the Southern Ocean is not static. It moves, and it does not always move in the same direction as the surface. Eggs falling into deeper layers can be transported differently from surface water, which in some regions may help return larvae toward productive areas rather than flushing them out into open ocean. How much this matters and where it applies is an active area of work. Either way, the strategy has an obvious dependency, and it is a hard one. It requires deep water underneath. If eggs sink and need distance to develop before hatching, then spawning has to happen where there is enough depth beneath the animals for the whole process to complete. Over shallow shelf areas, the geometry changes. Eggs can reach the seafloor, which is a different environment with different odds. Where krill spawn successfully is therefore constrained by bathymetry, as well as by food and temperature, and it requires the currents to cooperate. Everything about this cycle assumes that eggs sinking in one place and larvae rising days later arrive somewhere they can survive. The surface layer they come back to has to be productive, or reachable from productive water. If circulation carries the developing larvae into a region with no food, the climb succeeds and the animal starves anyway. That is the trap sitting under the elegance. The strategy is not just a behavior. It is a behavior tuned to a particular physical arrangement of depth, current, and productivity. It works where those things line up. Change the arrangement, and the same behavior produces a failure. The animals do hedge, and they deserve credit for it. A female does not spend everything in one release. Across a season, she can produce more than one batch, spread over weeks, putting eggs into the water under different conditions instead of bedding the whole year on a single day. That is a real defense against bad luck. It is not a defense against a bad season. Spreading eggs across 6 weeks helps when 1 week goes wrong. It does nothing when the summer is short, the water is wrong, or the adults never reached breeding condition in the first place. Now stack this with what came before, because the picture only makes sense assembled. The adults survive winter by shrinking, burning reserves, and cutting metabolism. But that suppresses their reproductive condition, so a bad winter can delay or reduce spawning even among survivors. The eggs that do get spawned drop into the deep, and the larvae must climb back on a fixed fuel supply, with no room for error in timing. The larvae that make it to the surface then face their own first winter, and their survival depends heavily on finding sea ice, with algae on it and structure to shelter in. 3 separate bottlenecks in sequence, each one dependent on conditions being right at a specific time in a specific place. That is why krill abundance swings so violently between years. It is not one variable. It is a chain of narrow windows, and the chain is only as good as its worst link in any given season. It also explains something about recovery that matters enormously later. If krill numbers drop sharply in a region, the population cannot simply refill from the survivors the way many marine invertebrates can. Rebuilding requires successful spawning, which requires animals in good enough condition, in the right water, over sufficient depth, with the currents working, followed by a productive summer, followed by a winter with enough ice for the larvae. Every one of those has to happen in sequence. A single strong year class can carry a population for years, but strong year classes are not reliable, and a run of poor ones cannot be compensated for by the adults simply trying harder. and none of that sequence can be hurried. Krill do not reach breeding size in a single summer. An animal spawning this season is the product of several years of conditions lining up, which means a region emptied of krill is waiting on a queue that has to be rebuilt from the front of the line. Some honest boundaries on this. The broad pattern of egg descent and developmental ascent is well established for Antarctic krill and has been described in detail. The specifics: how deep eggs go in different regions, how much variation exists in the timing, how larvae navigate the climb, what fractions survive it, vary between studies and locations, and researchers continue to work on it. Krill also show more flexibility in spawning location and timing than a simple description suggests. Treat the mechanism as solid and the details as a field in progress. What is not in doubt is the shape of the thing. This animal begins life by falling into darkness and then has to earn its way back to the light on a single tank of fuel before it has ever eaten anything. And here is the part that reframes it. Every one of those climbing larvae in every spawning season across the entire Southern Ocean is climbing toward the same destination, the productive surface layer where everything that eats krill is waiting, which is the point where this stops being the story of one animal's life cycle and becomes the story of the whole ocean because of what is stacked up there. The list of animals waiting at the top is not long. That is the surprising part. It is short. Startlingly short for an ocean that supports the largest animals in the history of life on this planet. And a short list is not a sign of strength. Start with a fact that should not add up. The largest animal that has ever existed lives here, not in the tropics, not in the richest coastal upwelling zones, in cold water at the bottom of the world. Blue whales, fin whales, humpbacks, animals whose energy requirements are difficult to picture, sustained in an ocean that spends much of the year dark and much of its area barren. Somewhere in the accounting, the Southern Ocean is doing something other oceans cannot. It is not producing more. Global productivity maps do not show the Southern Ocean as the planet's most fertile sea. Much of it, as covered earlier, sits blue and iron-starved. The advantage is not raw output. The advantage is the shape of the chain. Look at a typical open ocean food web anywhere else. Phytoplankton get eaten by tiny grazers. Those get eaten by slightly larger animals. Those get eaten by small fish, which get eaten by larger fish, which get eaten by something larger again. 5, 6, sometimes more steps between sunlight and a top predator. Each of those steps is a tax. The general rule in ecology is that most of the energy at any level does not make it to the next one. The exact figure varies by system and by species, but the direction never changes. A large fraction is lost at every transfer, burned on metabolism, movement, and the simple cost of being alive. Chain the steps together, and the loss compounds. By the time energy has passed through 6 levels, what arrives at the top is a small remnant of what was captured at the bottom. That is why big open ocean predators are relatively rare. They are living on the far end of a long series of subtractions. Now the Southern Ocean, phytoplankton, krill, whale, 3 steps. That is the whole path, in the most direct version of it, from sunlight to the largest animal in Earth's history. The energy captured by cells too small to see passes through exactly 1 intermediary before arriving in an animal that can weigh over 100 tons. 1 tax instead of 5. That is the entire explanation for what seemed impossible a moment ago. The Southern Ocean supports giants not because it produces vastly more energy, but because it wastes so little of it getting there. And the reason it can skip all those intermediate steps is the animal in the middle, because krill do the concentrating job that would otherwise require a whole ladder of successively larger organisms. Everything from earlier converges here. The feeding basket that strains microscopic cells out of the water, the swarming that packs those bodies into dense, findable concentrations, the high fat content that makes each mouthful worth taking. Those three traits together make a krill swarm the only thing in this ocean that lets an enormous filter-feeding predator make a living. A whale cannot eat phytoplankton. The particles are too small and too dilute. It would starve while filtering. It needs the energy pre-concentrated into packages of the right size, in the right density, in a predictable place. Krill are that service. The chain is short because one animal collapsed the middle of it. Look at what that made possible on the other side of the transaction. A baleen whale is, mechanically, a filter wrapped around a stomach, plates of keratin hanging from the upper jaw, fraying into a mat that holds back anything larger than the gaps between the fibers. It is the same solution as the krill's feeding basket, scaled up by an absurd margin, and it exists for the same reason. When your food is small and spread out, the winning move is to process volume instead of chasing individuals. Two animals at opposite ends of the size range, running the same trick, one of them only able to exist because the other one runs it first. Now look at everything else that eats them, because the list is where the real point is. Crab-eater seals, whose name is a mistake. They eat krill almost exclusively, and their teeth interlock into a strainer for filtering them out of the water. Fur seals, leopard seals in part, adelie, chinstrap, and gentoo penguins, feeding krill to chicks at colonies that number in the tens of thousands. Flying seabirds working the surface, Antarctic silverfish, and other mid-water fish, squid. That is not a diverse assemblage feeding on a diverse prey base. That is a large collection of very different animals, mammals, birds, fish, cephalopods, all funneling down onto the same species. And here is where it stops being impressive and starts being alarming. The short chain is efficient. It is also the definition of low redundancy. In most ecosystems, if one prey species declines, predators shift. Something else fills in. A fish that eats several kinds of prey adjusts its diet. The web has slack in it, because it is a web. Many nodes, many connections, alternative paths through. The Southern Ocean does not have a web at the critical point. It has a bottleneck. If krill decline sharply in a region, what does a crab-eater seal switch to? What does a chinstrap penguin feed its chick? What does a blue whale, whose entire feeding apparatus and body plan are built around engulfing dense swarms of small crustaceans, eat instead? The answer is not obvious, and for some of these animals, there may not be one. There are other prey in the Southern Ocean, other krill species, fish, copepods, squid, and some predators do use them, but nothing else combines the traits that make Antarctic krill work: the abundance, the density, the size, the fat content, and the reliable formation of concentrated swarms. Efficiency is the fragility. It is the same trait viewed from two sides. A long, wasteful food chain is robust because it has alternatives at every level. A short, efficient one is productive precisely because it does not. Now add the geography, which makes it worse. Many krill predators are not free to follow the food. Penguins breeding on land are tied to a colony, and during the chick-rearing season, an adult can only forage within a limited radius before it has to return. Fur seals with pups on a beach face the same constraint. These animals are anchored to a fixed point on a map for the most demanding weeks of their year, which means it is not enough for krill to exist somewhere in the Southern Ocean. They have to exist in specific places, at specific times, within reach of specific colonies. A shift of a few hundred kilometers in where the swarms are is nothing to a whale. To a penguin with a chick waiting on shore, it can be the difference between a successful season and a failed one. That is what makes the situation genuinely tight. The system's productivity rests on one species, and the accessibility of that species is constrained by breeding geography that cannot move. Some honest limits. Antarctic krill are not literally the only prey. Regional differences matter. Some predators are more flexible than others, and there are parts of the Southern Ocean where fish or other species carry more of the load. Ecologists debate how completely dependent particular predators are and how much substitution is possible under stress. The dependence is a matter of degree, not an absolute. But across the regions where krill dominate, the degree is high enough that nobody treats it as a minor factor. Krill-dependent predator populations are monitored specifically because their breeding success responds to krill availability, which makes them a signal of what is happening in water nobody can survey properly. Those programs exist for a practical reason. The predators are easier to count than the prey. Nobody can survey a swarm under winter ice, but you can stand at a colony and count nests, weigh chicks, and record how many of them fledge. The animals become instruments. So here is where this leaves the story. An animal, the length of a finger, sits at the junction of one of the shortest, most efficient food chains on Earth. It is the reason the largest animals in the history of life can exist in cold, iron-poor water. It supports mammals, birds, fish, and cephalopods simultaneously, with almost nothing standing in reserve behind it. That is a genuinely strong case for the animal mattering. It is also still a case about one ocean at the bottom of the world. The title of this story makes a much larger claim: the entire ocean, not one region, however dramatic. And if this ended here, that claim would be overstated. A Southern Ocean story wearing a global headline. So it has to be tested. Does anything that happens down there reach the rest of the sea? Does a swarm of small crustaceans below the Antarctic convergence have any connection at all to a fishery thousands of kilometers north, in warm water, in an entirely different hemisphere? The answer runs through the way the ocean physically moves. And it means the Southern Ocean is not the bottom of the world at all. Look at a globe from underneath, and the geography changes meaning. From above, the Antarctic looks like a terminus, the far end of every map, ringed by the coldest water on the planet, closed off by a current that circles the continent without interruption, a cold room at the bottom of the world, isolated by distance and temperature from everything that matters. That is the intuition, and physically, it has some truth to it. The Antarctic circumpolar current does run continuously around the continent, and the temperature and density change where cold Antarctic surface water meets warmer water to the north is a real boundary, a front that many species do not cross. Antarctic krill are among them. Their range effectively stops there, which is a large part of why it is so tempting to treat all of this as a closed local story, with no bearing on anywhere else. But surface geography is not the whole ocean, and the boundary that stops animals does not stop water. Here is what the Southern Ocean actually is. It is the place where the ocean's deep circulation turns over. Water that has spent a long time in the abyss comes up there, and water from the surface sinks there and spreads outward into the world's deep basins. It is a mixing zone at planetary scale, and it links the Atlantic, Pacific, and Indian Oceans into a single connected system, rather than three separate ones. Two movements matter for this story. The first is upwelling. Deep water rises toward the surface around Antarctica, and that water is loaded. Nitrate and phosphate accumulated over long periods as sinking material was broken down at depth. This is the reason the Southern Ocean surface is so nutrient-rich in the first place, and the reason the barren blue patches are such a puzzle until you know about iron. The nutrients arrive from below in quantities that far exceed what gets used. The second movement is the one that carries the story north. Surface water in the Southern Ocean sinks, and one of the water masses formed there spreads northward at intermediate depth, beneath the warmer surface layers, traveling into the Atlantic, Pacific, and Indian basins. It moves for a very long way, and a very long time before it comes back up. And when it does come up, in the low-latitude regions where deep water returns toward the surface, it brings its nutrient content with it. This is the connection. The nutrients that were in that water when it left the Southern Ocean become thousands of kilometers away and long afterward, part of what supports productivity in other parts of the world's ocean, which means what happens to those nutrients before they leave the Southern Ocean surface is not a local matter. Follow the logic carefully, because this is the point where the title either earns itself or does not. The Southern Ocean surface receives far more nitrate and phosphate than the organisms there can use, because iron limits how much growth can happen. Unused nutrients stay in the water. When that water sinks and heads north, it carries the surplus with it. Now change the amount that gets used locally, and you change what gets exported. If more growth happened in the Southern Ocean surface, if more nutrients were consumed and packaged into organisms, and then a share of that material sank locally into the deep rather than traveling north at intermediate depth, the water heading north would carry less. Productivity elsewhere would have less to draw on when that water eventually resurfaced. If less growth happened, the opposite. More surplus traveling north, more available at the far end. The Southern Ocean is a valve on the nutrient supply to a large portion of the world ocean, and biological activity in its surface layer is part of what sets that valve. This is a real and well-developed line of thinking in oceanography, and the mechanism is understood. What is not settled is magnitude. What fraction of low-latitude productivity depends on this pathway versus local recycling and other sources, and how sensitive it is to changes in Southern Ocean biology. Estimates exist, and they are argued about. Anyone who gives you a confident single number is overselling it. The mechanism, though, is enough to place krill in the picture. Everything covered earlier now reads differently. The daily migration that pulls material down out of the surface layer. The dense fecal pellets that sink fast enough to reach genuine depth instead of being recycled near the top. The molts dropping through the water column. The iron held in living tissue in the sunlit layer instead of sinking away. The grazing that converts dissolved nutrients into particles. Every one of those processes affects what stays at the surface, what goes down locally, and therefore what is left in water heading north. There is a further piece that rarely gets mentioned, and it comes out of what these animals eat. Diatoms build their cell walls from silica. Krill pack those walls into dense pellets and drop them. Silica pulled out of Southern Ocean surface water, that way is silica not traveling north. And the availability of it helps decide which kinds of plankton dominate in a given region. Change what happens to it down here, and you are nudging the composition of plankton communities elsewhere, not only the quantity. Krill are one of the largest biological influences on that balance in that ocean. They do not travel. They will never leave the Southern Ocean, but they act on water that does. That is the answer to the geography problem. Krill affect distant seas without going anywhere, by acting on the medium rather than moving through it. Two directions at once, which is worth stating plainly because it is easy to garble. They send material down. Pellets, molts, bodies, carbon, silica, nutrients, into deep water where it is out of circulation for a long time. And they hold material up. Iron in tissue, released back into the surface through excretion, molting, and being eaten, keeping it available in the light rather than letting it sink. What they stop from sinking and what they make sink are both part of the same accounting, and both feed into what the northward flowing water carries. Now add the carbon side, because it operates on the same physics. Carbon dioxide dissolves more readily into cold water, which means the Southern Ocean surface is a significant point of exchange between the atmosphere and the sea. It is one of the more important regions on the planet for that exchange. Some of the carbon entering there gets carried into the deep by physical circulation. Some gets carried by biology, the sinking of organic material, in which krill-produced particles play a substantial role. Nobody can give you a trustworthy figure for what fraction of that is attributable to krill specifically. The measurements are regional, the process is patchy, and estimates vary widely. What can be said is that the biological pathway is real, that fast-sinking krill pellets are among the more effective particles in it, and that any full accounting of Southern Ocean carbon export includes them. So the title's largest claim now has a foundation under it. Not because krill live in every ocean. They do not. Antarctic krill live in one region and never leave it. The claim works because that region is where the global ocean's deep circulation turns over, and because the biological activity in its surface layer influences what the outgoing water carries. The Southern Ocean is not a dead end. It is the junction, and krill are the dominant biological actor at that junction. Keep the honesty tight here, because this is the chapter where overclaiming would be easiest. Nobody is saying that if krill declined, tropical fisheries would collapse next season. The timescales involved in this circulation are long. The water takes a very long time to make its journey. Nobody has produced a reliable quantitative prediction linking krill abundance to productivity in distant oceans. And anyone who did would be working several steps beyond what the data supports. What can be said is this: the mechanism connecting Southern Ocean surface biology to the wider ocean is understood and accepted. Krill are the largest biological presence in that surface layer. Change them substantially, and you are changing an input to a system that reaches everywhere. On a timescale nobody would notice until long after it mattered. That is not a small claim. It is a claim with a long fuse, which brings this story to the present, and to the uncomfortable question that has been building since the first chapter. Everything so far has described how the system works. The concentrating step, the pump, the iron loop, the ice nursery, the short chain, the junction. None of it addresses what is happening to the animal right now. And what is happening is not one threat. It is several, overlapping in the same water. And what they do together is not the sum of what each would do alone. Ask what threatens Antarctic krill, and almost everyone gives the same answer. Fishing. There is a fishery, it is real, it has grown, and it targets exactly the swarms this story opened with. Vessels find dense concentrations with the same acoustics researchers use, and the most modern method, continuous pumping, the catch drawn aboard without hauling the net, removes the last natural break on how long a vessel can stay on a single swarm. The product goes mostly into aquaculture feed, pet food, and krill oil supplements sold on the basis of their omega-3 content. Now the part that complicates the obvious answer. The fishery is regulated, and by international standards, the regulation is unusually cautious. The body responsible sets a catch limit for the region where most of the fishing happens, and the actual catch has historically run at a fraction of that limit. Measured against the estimated biomass, with all the uncertainty in that estimate discussed earlier, the total removal is a small percentage. So on the numbers, the fishery does not look like the crisis. That is the honest reading, and it is also where the real problem hides, because the total is the wrong thing to look at. Fishing does not happen evenly across the Southern Ocean. It concentrates. Vessels work where krill are reliably dense and where conditions allow, and that means effort piles into a limited number of areas, particularly around the Antarctic Peninsula and nearby island groups. Those are the same places where krill-dependent predators breed. Go back to the constraint from earlier. A penguin with a chick on shore can only work a limited circle of ocean before it has to turn back. A fur seal with a pup has the same limitation. During the weeks that determine whether a breeding season succeeds, these animals are anchored to a point on a map and must find krill nearby. Now put a concentrated fishery in that same radius during that same season. The catch can be a small share of the ocean-wide total and still remove a meaningful share of the krill accessible to a particular colony at the exact time it matters. A regional total that looks conservative on paper can mean something very different in a specific bay in a specific month. This is understood by the people managing it, and it is why the debate is not really about the overall limit. It is about how catch is distributed in space and time. There have been efforts to spread effort out and to protect areas near predator colonies, and there is ongoing argument about how well those measures work and how they should evolve. Some are voluntary, some have expired or been contested. This is live, current, and unresolved. There is a wrinkle in the timing as well. For most of its history, the fishery worked in summer, because that was when conditions allowed. Better vessels and onboard processing have made winter operation more practical, which pushes the pressure into the months when the population is least observed, least understood, and already running on reduced reserves. Nobody can survey what is happening underneath that, which means catch is being taken legally and inside the limits out of a season nobody can measure. So the first correction, the risk, is not the tonnage. It is the geography. The second correction is larger. Fishing is one pressure among several, and it may not be the most consequential. Take the ice. Everything from the nursery chapter applies directly. Larval krill depend on sea ice for winter food and shelter. Antarctic sea ice has behaved in complicated ways. It does not track the Arctic. Regional patterns differ, and it has surprised researchers repeatedly. But recent years have included extents well below anything in the modern observational record, and the region around the Antarctic Peninsula has seen substantial changes. That region is also one of the most important krill areas, and one of the most heavily fished. The mechanism does not require a forecast. Fewer months of ice, thinner ice, or ice that forms late and retreats early means less winter grazing surface and less structural shelter, in the season when the youngest animals have no alternatives. Poor ice years plausibly mean poor recruitment years, and recruitment is what carries this population. It also does not need to be dramatic to matter. Ice that arrives 3 weeks late is still ice. It is simply 3 weeks of missing food at the point in the year when the smallest animals have the least margin. And repeated often enough, a run of unremarkable winters produces a gap in the generations that nobody notices until the animals, which should have replaced the current ones, fail to show up. Then, temperature. Krill are cold water animals. Their distribution has been reported to have contracted southward in parts of their range over recent decades, while this a finding that has been debated because of the survey and sampling problems described earlier, but which has enough support to be taken seriously. Here is why southward contraction is worse than it sounds. Moving north, there is open ocean and room. Moving south, there is a continent. The habitat available shrinks as the animals retreat toward it, and the areas they retreat from are precisely where many predator colonies are established. A colony cannot follow. The birds and seals are tied to ground that does not travel south with the krill. Then the water chemistry. The ocean absorbs a share of the carbon dioxide added to the atmosphere, and that changes its chemistry, lowering pH. Cold water absorbs carbon dioxide more readily, which means polar oceans are affected earlier and more strongly. Laboratory work has found that krill eggs in early developmental stages are sensitive to increased carbon dioxide in the water, with reduced hatching success at elevated levels. Be careful with that finding. It is laboratory work, using specific conditions and extrapolating from a tank to a life cycle that involves eggs sinking 1,000 meters through varying water is not straightforward. The relevant conditions vary with depth and region, and the animals may have responses that experiments do not capture. What it does establish is a mechanism worth watching, and it targets the stage that already has the least margin, the one falling into the dark with no defense and no reserves. Now, the actual point of this chapter, because the individual pressures are not the story. They arrive together in the same waters, aimed at different parts of the same life cycle, and they interact. Fishing concentrates where predators breed. Ice loss is strongest in some of those same regions. Warming pushes the animals south into a shrinking area, which concentrates the remaining population, making it easier to find and easier to fish, and more dependent on fewer places working. That is not addition. Each pressure changes what the others do. A population reduced by a poor recruitment year is more affected by a given tonnage removed. A population squeezed into a smaller area is more exposed to local depletion. A larval stage weakened by chemistry has less tolerance for a poor ice winter, and the animal's own survival tools, shrinking, reserve burning, metabolic reduction, all assume a good season arrives afterward to rebuild from. They are gap-crossing strategies. They are not strategies for a permanently worse baseline. So the failure mode here is not a crash. Nobody expects the Southern Ocean to go from full to empty in a season. It is a slide. Recruitment failing more often. Distribution tightening. Local depletion in the wrong places at the wrong times. Predator breeding seasons failing more frequently than they succeed. A system that still contains krill, still produces swarms, still shows up on sonar, but no longer delivers reliably where and when the animals, depending on it, need it. And detecting that slide requires exactly what this story established is hardest: long-term abundance data in an animal that is patchy, hard to count, hard to age, and living under ice for the months that matter most. There is one more feature that makes this difficult to manage rather than merely difficult to study. Every pressure listed here runs on a different clock. A fishing season is measured in months. An ice trend takes decades to confirm. Ocean chemistry moves slower still. Decisions get made on the fastest of those clocks, and the consequences arrive on the slowest. The honest position, stated plainly, is this: krill are not on the edge of extinction. The population is large, but the pressures are documented. They overlap in the most important regions. The animal's dependencies are narrow at several points in its life cycle, and our ability to see the trend clearly is limited. That is enough to make the question worth asking properly. So run it forward. Not as a prediction. Nobody can predict this, and inventing numbers would be worthless. As a thought experiment built only from mechanisms already on the table, what does that ocean look like without them? Everything from here is a scenario, not a prediction. No dates, no numbers, no forecast, just the mechanisms already established, followed to where they lead. Suppose Antarctic krill decline substantially across their core range. The intuitive picture is emptiness. Cold water with nothing in it, a sea that has been switched off. That picture is wrong, and the reason it is wrong is the most unsettling part of this whole story. The sunlight would still arrive. The nutrients would still upwell from below. The iron would still be delivered at the ice edge and around the islands. Algae would still bloom. The energy entering that ocean would not decrease at all. Something would still eat it. The Southern Ocean already contains an alternative grazer, and it is not remotely like a krill. Salps are gelatinous animals, barrel-shaped, transparent. Closer in ancestry to vertebrates than to crustaceans, though nothing about them looks it. They pump water through an internal mucus mesh and strain it. And that mesh catches particles far smaller than a krill's feeding basket can hold. They can form chains of connected individuals, drifting through the water in translucent ropes, and they reproduce in a way that makes krill look ponderous. Under good conditions, salps can multiply explosively, producing dense blooms in a fraction of the time krill need to build a generation through spawning, sinking eggs, a developmental ascent, and a winter under the ice. Here is the part that matters. Salps and krill do not favor the same conditions. The general association reported in the Southern Ocean is that krill do better in colder, higher ice conditions, while salps tend to be found in warmer, lower ice water. And that in some regions and periods, Salp presence has increased where krill have not. Treat that carefully, because it is one of the more contested areas in this field. The pattern has been reported and debated. The mechanisms are not fully resolved, and how far it generalizes across the Southern Ocean is genuinely argued about. It is not a demonstrated replacement. It is a documented tendency that would be reasonable to expect to strengthen under warmer, lower ice conditions. But as a mechanism to reason with, it is enough. Because if grazing shifted substantially toward gelatinous animals, the ocean would not empty. It would fill with the wrong thing. And the way a salp handles food is not a reduced version of what a krill does. That mucus net catches particles far finer than a feeding basket can hold, which means salps can graze a plankton community krill would struggle with, including the smaller cells that warmer, more stratified water tends to favor. That is not a coincidence. It is part of why the two animals suit different oceans. So this would not be a system limping along on a poor substitute. It would be a system working perfectly well on different terms, with a different set of winners. Now work through what that does, using everything established. Start with the food chain and the elegance that turned out to be fragility. A salp is mostly water. Its energy content per unit volume is far below a krill's. No thick lipid reserves, no dense muscle. Nothing like the fat content that makes krill worth an enormous animal's effort. And they do not swarm the way krill do. They bloom and drift, in a distribution that does not offer the same dense, reliable, targetable concentrations. A blue whale is built around one specific task, engulfing a large volume of water containing a dense mass of small, fatty crustaceans, and doing it efficiently enough that the energy gained exceeds the enormous cost of the lunge. That equation has no slack in it. Replace the target with a low-energy gelatinous animal in a diffuse distribution, and the arithmetic stops working. Not because the whale would refuse to feed, because the return would no longer cover the cost. The three-step chain would lengthen. Energy would move through more intermediate steps or into pathways that lead somewhere other than large predators, and each transfer takes its share. The ocean would still be productive. It would stop being able to afford giants. The predators anchored to land would fail first, and they would fail fastest. A whale can search. It can move hundreds of kilometers to find better water. A penguin with a chick cannot. A fur seal with a pup cannot. Those animals must find dense food within a limited radius of a fixed point during the weeks that decide the season, and there is no substitute available within that radius. The visible signal of this scenario would not be a dead ocean. It would be colonies. Reduced breeding success, then fewer birds returning, then thinning ground where dense colonies used to be, in an ocean that still looks full when you sample it. Then the pump. Salps produce fecal pellets, and some of them sink well. Salp blooms can export carbon effectively, but the process is different. Different particle characteristics, different timing tied to bloom cycles rather than a nightly migration, and without the vertical transport that a migrating animal performs with its own muscle. The krill pump is specific. Feed at the surface at night, carry the load down hundreds of meters by dawn, release dense, fast-sinking pellets already deep. That is a mechanism, not a generic function. And it is not obvious that anything else replicates it at the same scale. So the carbon pathway would not stop. It would change character. And a change in character at the junction where the global ocean turns over is not a local adjustment, then the iron. Krill hold iron in living tissue in the sunlit layer. Predators eating krill release it back into the surface. That loop keeps the scarcest resource in that ocean circulating in the light rather than sinking away. Take out the animal that concentrates it and reduce the predators that recycle it, and the loop opens. Whether a salp-dominated system holds iron in the surface layer, The same way is not established. The mechanisms are different enough that assuming equivalence would be careless, and recovery would be slow, for reasons already covered. Krill cannot simply refill a region. Rebuilding requires adults in good enough condition to spawn, in water deep enough for the eggs to develop, with currents delivering larvae to productive water, followed by a winter with enough ice for those larvae to survive. Every link in sequence. Salps, in warmer lower ice water, would rebuild far faster. That asymmetry is what makes the scenario stick rather than swing back. Here is what the whole thing amounts to. Collapse is the wrong word for what people picture when they hear it. This would not be an ocean gone quiet. The water would be full. Blooms every summer. Grazers in enormous numbers. Particles sinking. Life everywhere. It would simply be an ocean that had lost the ability to concentrate energy, and therefore an ocean that could no longer build anything large out of it. Not empty, shallow. That is the reframe worth keeping. From the beginning, this was never a story about an animal being important because a lot of things eat it. Krill are not a food item that happens to be abundant. They are a mechanism. They are the step that takes sunlight captured by cells too small to see and packs it into something dense enough for the largest animals in the history of this planet to live on. They are the step that takes carbon from the surface and drives it into deep water using nothing but muscle and the fear of being seen in daylight. They are the step that holds a trace metal in the light instead of letting it fall. Take the mechanism out and the ocean keeps running. It just runs at a smaller scale, with more steps, less transfer, and nothing at the top big enough to notice from a ship. And none of it would announce itself. There would be no year in which the Southern Ocean visibly changed hands. The shift would be recorded if it were recorded at all. In breeding numbers from a handful of colonies and in survey figures everyone already knows are uncertain. So go back to the beginning. A research light on black water. A single translucent animal drifting through the beam. Glassy shell, black eyes on short stalks, a green stripe of algae down the middle of it, 10 small lamps along its belly, paddles beating without stopping, because stopping means sinking. Around it, in the dark the beam does not reach, are millions more, holding formation, waiting for the light to fail so they can climb. Then the beam moves on, and the whole thing goes back to being invisible. That is where this ends. The animal that everything down here depends on is the one we can barely see, cannot properly count, and only started taking seriously once we noticed what happens when it is gone.